And here we are in our last little recorded lecture for chapter 12. We are just going to wrap up with what happens once our nerve signal has reached the end of our axon, how we actually communicate to our other cells, and then I want to circle back around, put all of our pieces together, and think about the nervous system as a whole. Next week, we will have an exam. It'll be on chapters 10, 11, and 12, so muscle anatomy and physiology, and then some of those major muscle groups that we talked about, and then the introduction to the nervous system. We can then move into brain anatomy, spinal cord anatomy, peripheral nervous system responses, a whole slew of other fun things for the nervous system. We are going to actually start where our nerve signal ends. So a nerve signal is going to end once it reaches the synaptic knob at the end of our axon. Once again, we've already talked about this particular anatomical feature. We've talked about it between neurons and muscle cells. Here we're going to flip the script a little bit and we're going to talk about it as if two neurons are communicating with each other. When we have two cells communicating with each other, one of them is going to be the presynaptic cell. One of them is going to be the postsynaptic cell. So a presynaptic cell. Is going to be the cell through which the nerve signal is moving and the cell that is doing the secretion. So it is going to be secreting our neurotransmitter. The neurotransmitter is that chemical signal. The chemical signal or the neurotransmitter is going to move across our synaptic cleft, just like we saw with our muscle cells. The cell that is receiving the neurotransmitter stimulation is postsynaptic. And so it is going to respond to the neurotransmitter. Let's identify the pieces of our synapse. First of all, I'll show you here is the axon of our presynaptic neuron. Here are going to be where we have our synaptic knobs. They are communicating with the dendrites. Remember, the dendrites are where most cells are going to synapse or where most neurons are going to receive incoming communications. We do also get synapses with the soma sometimes, and also sometimes with the axon, but most commonly with the dendrites. So the dendrites receive that information. Those are going to be our local potentials. Local potentials sometimes will move all the way to that axon hillock, and then will cause action potentials to hop down our axon as a nerve signal. So here, sort of zoomed in, is our presynaptic neuron. So this would be the axon. This is going to be the synaptic knob. Inside of the synaptic knob, we have our synaptic vesicles. Those are going to contain our neurotransmitters. Some of these synaptic vesicles are going to be sort of close to the end here, so that they can actually just easily sort of undergo exocytosis and secrete their neurotransmitters into our synaptic cleft. So just like we saw with muscle cells, these are functional, not physical connections. So we don't touch cells here. Our neurotransmitters are essentially open or released into sort of an open space. They will diffuse from the presynaptic neuron to the postsynaptic neuron. In the postsynaptic neuron, we are going to have ligand-gated channels, which will open and allow for our local potential to begin. Most of the time, the synapses are going to be chemical, so that's our neurotransmitters. It was originally thought that synapses carried electrical responses from cell to cell or electric signals from cell to cell. That is actually true in some parts of the brain and areas where we have ions that move through gap junctions. Not true for most of the nervous system. So most of the time, communication between a presynaptic and postsynaptic neuron are chemical in nature, and it's those neurotransmitters that do move across our cleft here. I think I sort of said this. Our action potential is going to reach the end of the axon at that synaptic knob, that little broad area. Sort of expands at this area. And then we're going to get exocytosis of the neurotransmitters. They will then move across the cleft, bind to their ligand-gated channels in that postsynaptic neuron membrane, and then result in either depolarization or hyperpolarization. Up here, this is sort of a standard way to draw neurons. We will continue to see this sort of drawing. Sometimes we'll also see even a little bit simpler than that. We'll just see this. And so that would be sort of showing us, this would, whoops, not the eraser. This would be the soma and the axon, and this would be the synapse between the presynaptic and postsynaptic cells. So that's what this is showing us, the soma, the axon. This is presynaptic. There's our synapse. The synapse is going to form with the dendrites, which would be associated with the soma of our postsynaptic cell, and we're always going to get information moving in that direction. There are different types of receptors that are going to respond to our neurotransmitters in different ways. The one that we've talked about most are called ionotropic receptors, and so these are our ligand-gated. When we have these types of receptors, our neurotransmitters, which are shown here in red, are actually going to bind into sites on this surface of this particular receptor and cause it to open. We sometimes have metabotropic receptors as well. These don't open like channels. Instead, what happens is our neurotransmitters bind to the outside. We get sort of a conformational change, and we activate a metabolic response inside of the cell. So we get a different type of response internally than we see with our ionotropic receptors. If you go to your textbook, you'll see that there is a table that lists a whole bunch of neurotransmitters. There are more than 100 different neurotransmitters described. They fall into a number of classes. So acetylcholine is a class of its own. We also have amino acid neurotransmitters, monoamine transmitters, neuropeptide transmitters. These are different categories or classifications of neurotransmitters. I'm not going to get real deep into all the differences here. You can kind of see some of those differences just by looking at them. Our amino acid neurotransmitters here are just going to be an amino acid, basically. Monoamines, so this would be one or two amino acids, pretty much one amino acid. And then here neuropeptides are strings of amino acids, so there's sort of different varying lengths and levels of complexity here. I just wanted to show that these are different types and classifications of neurotransmitters. You can look at this table. So this is a link. If you open the PowerPoint, you can just go to your textbook and find that table. But it'll show you the neurotransmitters. These are molecules. Let's actually define neurotransmitter while we're at it. Our neurotransmitters are going to be molecules that are synthesized by a neuron. And then they're also going to be released when a nerve signal reaches the axon terminal. Then they are going to traverse that synaptic cleft or go across that synaptic cleft and have a specific effect on our postsynaptic cell. So each of these neurotransmitters does something specific and actually changes the physiology of the postsynaptic cell. The last little thing that I want to talk about sort of combines the idea of neurotransmitters with what's going on when they reach the postsynaptic cell. So we know that our typical resting membrane potential is minus 70 millivolts and our threshold is minus 55 millivolts. Anytime we get a voltage change towards the threshold, we are increasing the chance of an action potential happening. So if that occurs, we call that an excitatory postsynaptic potential, which we abbreviate EPSP. Anytime we have hyperpolarization of the postsynaptic cell, we then decrease the chance of an action potential, and therefore we have an inhibitory postsynaptic potential, or an ISPS. That is to say, some neurotransmitters are going to excite the postsynaptic cell and make it easier to reach that action potential. Other neurotransmitters are going to inhibit the postsynaptic cell or make it more difficult for an action potential to occur. In general, an EPSP, so an excitatory postsynaptic potential, is weak. So one ESPS would cause a 0.5 millivolt change. We need about 30 ESPSs to actually reach our threshold. So to go from minus 70 to minus 55, we have to get 30 EPSP signals to a cell to reach that threshold. There are two ways for us to add together EPSP, sorry, I keep saying ESPS. EPSP. Those are either temporal or spatial. If we have temporal adding of our excitatory postsynaptic potentials, we have a single synapse that is generating EPSPs quickly. Okay, so we'll just use this one for example. This particular synapse, this neuron is sending a signal, boom, a signal, boom, a signal, boom. When we add those signals sort of rapid fire to this one area, that would be temporal summation or temporal addition of our excitatory postsynaptic potentials. Sorry, I got distracted. So excitatory postsynaptic potentials, when we add all of those together, we're more likely to get the cell to reach its action potential. Spatial, we said in the past, and I'll remind you now, that it is possible for one postsynaptic neuron to synapse with many presynaptic neurons, right? So in this case, we would have several synapses adding at once, right? So this is spatial. So here we actually have 4 synapses that are excitatory. Those 4 excitatory synapses are all sending excitatory postsynaptic potentials to the cell at once. Those 4 would add together and create a larger excitatory response. And so we can actually add excitatory and inhibitory together. And so I sort of want to trace this membrane potential down here. So here we are at a resting potential. We then get a couple of excitatory bumps. And so we get close to threshold, but as soon as we're getting close to that threshold, we actually get an inhibitory bump, which is going to open, I guess I should write that on here. The way that we sort of hyperpolarize our cells most often is going to be through opening of chloride channels. So we get sort of an increase in the anions that move into a cell. That is going to bring our potential back down or lower us into the negative area a little bit further. So we bump ourselves back down. We then enter sort of a neutral phase where nothing's happening. We bump ourselves back up with another excitatory postsynaptic potential. We get another inhibitor. So these are all sort of summing together, right? We add 2, we take away 1, we add 1, we take away 1, we add 2, we get to threshold, we get an action potential, and then we move back into our repolarization, hyperpolarization, and resting membrane potential. So summation, I realize I jumped into that without actually defining this well for us. Summation is going to be the adding up of postsynaptic potentials okay, so we could get a whole bunch of inhibitory postsynaptic potentials that would push this cell into hyperpolarization for a while, and then we might get some excitatory and push up a threshold, get an action potential, go back down. It all just depends on what messages are coming from all of these. Okay, so that is sort of the bulk of what I have for this lecture. I realize that in your lecture there's a couple slides on nerve anatomy. I'm actually going to move that to a later lecture and sort of leave this one where it is. So I have two sum slides. This one is telling us to put all of our pieces together and look at, sort of go back to where we started by looking at the whole nervous system and what the nervous system is doing. Right, so we're going to get a stimulus. That stimulus is going to be measured or monitored by some sort of receptor. In that receptor, we have sensory neurons. These are also called afferent neurons because they are moving a signal towards something. And so in this case, they are moving it towards the brain. So once this afferent fiber moves that message of environmental change into the nervous system, we have a whole bunch of interneurons which communicate with each other. They integrate the information. They make a decision on how to respond. That information is sent back out a motor neuron, and this is an efferent response because moving away from the brain, and that sends messages to our effectors. Effectors are either muscle or gland. And the effectors are going to carry out the decision that is made by the brain. So they'll adjust something and respond to the internal or external environmental change. So here is an unlabeled situation. And then, so there are 7 numbers on here. There are 7 terms that I'm asking you to label. So I will let you sort of work your way through this and figure out exactly what's going on. Remember, afferent, maybe I shouldn't give you this hint now, but equals sensory, and efferent equals motor. Okay, so that is it for this chapter. We will chat soon. Please let me know if you have questions, comments, concerns. Yeah, I hope all is still going well.